In this video we will discuss about assay condition for the optimum response from 8XNFAT reporter. So far we have optimized this 8XNFAT reporters in terms of the promoters that can be used before the reporter genes and the reporter proteins that gives us the optimum signals. So here they will check, or the researchers have checked what are the optimum conditions for the assay. So in order to check that, they used two different T cell receptors, that is GSE 6H9 or N1E6, and they used the APC cells that can express these receptors to the T cells. And along with that, they also use anti-CD3 to activate the T cells. So when the T cells are not activated, they see minimum signals for both the receptors conditions, and that is minimum signals in terms of this their reporters does not shows a green signals. And when they are stimulated with this insulin 923 using the GSE 6H9 or this 1524 using 1E6, they are able to see increase in the reporter signals, and this increase is usually up to 18 hours, and after that they see stagnations for both the cases. And this is also true for this anti-CD3 antibodies in both the cases. And thereafter they wanted to check the optimum number of cells, T cells, that can be used. So when they used around 2 to 10 to about 4 T cells, they see a minimum IL-2 productions compared to the other conditions, that is 1 into 10 to the power 5 and 2 into 10 to the power 5. However, the ZsGreen signals is high for the 2 into 10 to the power 4 cells per well, that can be seen in the panel E and also for the panel F. And when they check it for this anti-CD3, they can see that for the anti-CD3 also signals are high when 2 into 10 to the power 4 cells are used. So they have decided to continue with the 2 into 10 to the power 4 cells per well. And thereafter they checked this interleukin 2 productions and compare it with this ZsGreen positive cells, and they are able to see maximum signals for this 2 into 10 to the power 4 cells per well compared to the other conditions. And this is true for both the T cell receptors. Now previously they have shown that if this anti-CD3, the CD3 genes are present in the T cells, then it will stimulate much more faster. So they introduced CD3 genes, transduced CD3 genes in the T cells, and then check the expressions of those CD3 genes, and we can see that the CD3 genes are expressed, although they are already expressed in these T cells, and we can see that levels does not change significantly. And when they check the CD4 levels, they are also increased. They are increased with the addition of the CD3, that can be seen here. And for the CD8 levels, it goes down with the addition of the CD3, that can be seen here. But the CD28 levels remain same before and without additions of CD3. And thereafter they check the ZsGreen positive signals through the anti-CD3 antibodies and also through the chemical stimulator, that is PMA or inomycins. And with PNA plus inomycins, the signals with and without CD3 additions remain same. And for the anti-CD3 antibodies, with the anti-CD3 antibodies, we can see an increase in the stimulations. And these results are similar results they have observed for the I1E6 receptors also, when they use 1E6 T cell receptors. And thereafter they wanted to check, since the CD28 antibodies are also expressed evenly, they check the CD28 anti-CD28 antibodies results, and they can see that when they use anti-CD28 antibodies to activate the T cells, they are able to see its signals in the ZsGreen positive cells, And when they use both the things, when they use this insulin and anti-CD28 antibodies, they are able to see signals that can be seen here. And when they use insulin plus anti-CD3 additions to the cells plus CD28, the signals remain same. But when they use anti-CD3 antibodies, in that case the signals are high with the additions of CD3 antibodies, and also slightly higher with the additions of with the use of this anti-CD28 antibodies. And a similar kind of results they have seen for the T cells activations using 1E6 receptors. So this is how they optimize the conditions, that is 2 into 10 to the power 4 cells per well is optimum, and 18 to 22 hours are also optimum for the observations of the activation of the T cells. So this is what they have find out about the optimum conditions for the assay using this 8XNFAT reporters. So I hope you understood how they come up to their optimum conditions, and if you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.